Hi everyone, Tash, Yastashmas Science Tengku Jaafar Fully Residential School. Thanks for watching our video. So, we are here to story you about our project, which is Precus, the natural preservatives for chilled chicken ground meat. Refrigerated chicken can only last for 1 to 2 days compared to frozen chicken that can last a year, which means refrigerator will fast purge. Next, to prevent or delay the microbial growth, antimicrobial preservatives have been utilized for many years, such as butylated hydroxynitrate, which is very harmful for human health. Last, FICUS CARICA is one of food additives that exhibit excellent antibacterial activities against tested gram-positive and gram-negative. The usage of FICUS CARICA extract has been proven by various scientific studies as a food preservative, but there was no practice by using FICUS CARICA leaf. As we know, leaves are one of the agriculture waste products. Therefore, by using leaves as a preservative, we can minimize such wastage. We have been using 3 types of parameters to monitor the extract preservation, which are total viable counts, organoleptic testing, and physical-chemical property.
The main ingredient is FICUS CARICA leaves. So, how was the extract being prepared? Firstly, fresh fig leaves were collected and washed thoroughly using clean water. To increase the total surface area, the leaves were torn into small pieces, increasing the rate of evaporation during the drying process in a hot oven. Then, grind the dried fig leaves and filter them. In the sample preparation, fresh chicken meat that had been washed using distilled water will be divided into 3 groups, which are TO, T1 and T2. No preservatives were added to TO as control, while 1 gram and 2 gram of fig leaves extract were added to T1 and T2 respectively. Each sample was packaged in sealed bags and stored in the refrigerator. This sample was drawn on day 3 and day 9 and analyzed for different preservation parameters.
Now, let's move to the results. The results are all based on the parameters that we use to monitor the preservatives. Firstly, let us see the effects of the preservatives on the microbial population. As we can see on the first graph, TO has the highest reading of microbial power. It is because it doesn't contain any preservatives, whereas T2, that have the highest amount of preservatives, had an increasing number of microbes on day 9, but is still lower than the other 2 samples. Hence, it proves that these preservatives can prolong the shelf life of chicken meat in chilled storage. Next is the organoleptic test. Based on the second graph, we can see that T2 has scored the highest sensory point, whereas TO has the lowest. It's because TO has an unpleasant color and odor, whereas T2 has a better one than it. So, it proves again that FICUS CARICA is working as a preservative. Lastly is the pH value. Graph 3 shows that the pH value had an increasing trend throughout the storage periods in all 3 samples. But on day 9, we can see that T2 has a higher pH value than T1. It is because the last pH value increase might have been due to the liberation of ammonia compound as a result of endopractice activity or the proteolytic microbial flora presented in the raw meat.
As conclusion from the comparative study of natural preservatives, it was concluded that FICUS CARICA leaves could be utilized as antioxidant and antimicrobial in preservation of chicken ground meat.